welcome you to the second edition of the ICTP-IBM Brahmagupta AI Prize for Early Career Scientists. The prize was established in 2025 thanks to a generous contribution from IBM, which I would like to warmly thank them for this. And the aim of this prize is to promote research focusing on approaches to AI and science that can benefit society at large. The 2026 prize is awarded to Professor Michael Skinnider, currently assistant professor at Princeton University. We will hear the citation elaborated by the chair of the selection committee, Professor Mark Mezar from University of Bocconi. And within I would like to just say a few words that we think of this, our collaboration with IBM also in the context of the International Consortium for Scientific Computing, ICOMP, which is one of ICTP's strategic priorities. As you know, ICTP is committed to open science, to make advanced science globally available. And of course, it would be very natural to have artificial intelligence as part of that thinking in the years to come. So as a result, artificial intelligence is receiving increasing institutional focus. And in fact, this emphasis will be highlighted next year. Actually, we initially planned it as a semester, but it's almost looking a year in 2027 will be dedicated to artificial intelligence for scientific discovery. It will feature a number of dedicated AI-focused activities, culminating in June 2027 as the month of AI, alongside the new edition of the ICTP-IBM Brahmagupta AI Prize, and several colloquia and school lectures by distinguished AI scientists. Congratulations and welcome all of you to this today's program. And it is now my pleasure to leave the floor to Alessandro Curioni, a good friend of ICTP and Vice President, IBM Europe and Africa, and also Director of IBM Research Zurich. So, Alessandro.
Good afternoon, everybody, and it's really a pleasure to be here for this new edition of this AI Prize that, as Satish has said, we have established together a couple of years ago, together with another prize on quantum computing. And the reason why we have done it, because there are no doubt that IT has been always a super important instrument for scientific discovery and for science. But in the past couple of years, with the revolution that AI brought on the table and quantum is bringing on the table, this instrument is becoming much more powerful, right? And the impact that this instrument is going to bring to science and ultimately to society depends on the work that many people, like the young scientists are here in the room today, are going to do to create the right algorithm to exploit this instrument at best. And that is why I am very proud today also to have this prize to Michael, because one thing is the instrument, another thing are the algorithms to be able to make the instrument powerful, but then you have to use these instruments, right, to advance science if you want to make a difference. And especially advancing science in biomedical research, I do believe that is one of the quests that we have in front of us, that probably is most important for the society to try to allow each of us in an inclusive way, by the way, to live longer and to live better. Yeah? So Congratulations, Michael. Really, again, thank you for everything you have done and you will do in the future. And looking forward to hear your presentation later today. And thank you for all of you for being here today for this prize. Thank you again.
Mark connected. Can somebody? He's connected. You have Mark on the screen?
Yes.
Okay, excellent. Sorry, Mark. So the floor is now yours, Mark. Mark Mesar is a professor at the University Bocconi in Milan, and he was the chair of the selection panel for this year prize. So thank you, Mark, for accepting to say a few words and motivate. What motivated you to choose Michael Skinnider for the prize? Mark, the floor is yours.
Hello, everybody. It's a pleasure to be part of this meeting. I regret I cannot be with you physically, but I am here with the Zoom. It's a great pleasure to participate this jury of the ICTP-IBM Brahmagupta Prize. I think it's a great prize. ICTP has a wonderful tradition, has established very important prizes, for instance, like the Dirac Medal, which has become really a landmark for theoretical physics. And thanks to this collaboration with IBM, it was able to create last year this AI prize, which is a prize dedicated to outstanding early career researchers. Early career is, in this case, within 12 years of PhD, which is not that early, but leaves the time to have done a very substantial accomplishment, as is the case of our laureate today. The jury, AI is very diverse. AI is becoming more and more a kind of generic term with a lot of diversity. The prize wants to honor significant advances in AI theory, in algorithms or applications, and particularly in research that uses open and responsible approaches to advance science and benefit society. And this is, of course, something that we kept in mind. The jury is also diverse. The jury consists of Anima Anand Kumar from Caltech, working on machine learning for modeling complex physical systems. This year, Anima did not take place in the discussion because she had nominated a candidate. Then there is Ayman Kanjo, who works on more tiny machine learning intelligence for small devices. She's from Nottingham Trent University. Then we have Patrick McSharry from Carnegie Mellon, who works on predictive AI for predictive analysis, for decision making, for risk analysis. We have Andrea Montanari from Stanford, who works at the intersection of machine learning, high-dimensional statistics, information theory, and myself, who work on, let's say, the theoretical foundation of machine learning, basically. So this year we had a very important and very nice selection of candidates. We had very high-level candidates in diverse fields. There was a first screening done by the colleagues at ICTP, and then the jury decided to run in first shaping a shortlist and then making the final decision. So, as has been said, the final choice of the jury is for Professor Michael Skinnider. It's a great pleasure to congratulate him for this prize. Michael is from Princeton University, and he was given this prize, awarded this prize for pioneering the application of artificial intelligence to biomedical discovery and for developing transformative AI methods that reveal previously unknown chemistry, accelerate metabolite discovery, advance neuroscience, and address major challenges in human health. Through exceptional scientific creativity, interdisciplinary vision, and an outstanding record of innovation, Michael Skinnider has demonstrated how AI can profoundly expand the frontiers of scientific knowledge and accelerate its translation to societal benefit. So I think that, Michael, you have done, you are doing, and you will do certainly even much more absolutely wonderful work on AI, also on its application to this whole field of biomedicine. And this is a field which certainly, I always quote it as one of the major domain of application of AI for positive application. There are every day in the newspapers there are threatening applications of AI, and people sometimes forget all these benefits of AI that one can have. And I think you are really paving the way for this very important application. And the nomination, as you saw, has emphasized both the breadth and the importance and the depth of your contribution in this field. So it is really with all my personal admiration that I am very happy to award you this prize.
Thank you. Thank you very much, Mark, for your words and for your work, actually, as a panel, on behalf of the panel. It's now time for the award ceremony. So I would like now to call Alessandro Curioni and Atish, please. We're actually almost half an hour ahead of schedule, which is quite unusual for this type of events. So I just wanted to check if Professor Wishart is connected. He's actually connecting from the West Coast, so I'm not 100% sure he's connected. They're checking. Otherwise, what I would suggest, my friend, is can we perhaps do the group picture now? Where do we do it? We do it with people staying on their... I would suggest so, because otherwise it's a bit disruptive. No, we ask people to come downstairs.
We'll stand in the...
Yeah. Okay, so I would suggest that we have a slight change of program and we do the group picture now, because Professor Wishart is not yet connected. Unfortunately, he's, I think, 8 or 9 hours ahead of time, so it's early morning for him, so he's probably going to connect us the last minute. So, okay, let's do the group the group photo then. Are we going to... Okay, so try to perhaps move away from, try to fill the two aisles at the center so that we can take a picture from below. No? Okay. Yeah, we're 20 minutes ahead of schedule.
So what should we do?
Well, we just wait, because he's not connected. He's connecting from the West Coast. So, well, this is the...
And we write to him.
Well, but he's introducing the speaker. I think we'll have to just wait until he's connected. I'm afraid.
And then
This is the keynote talk, but it's a long talk, so it's... yeah. No, I guess we just wait a few minutes. Okay, apologies for this, but we're waiting for Professor Wisher to connect. Meanwhile, I can perhaps mention that the reason we organized the ceremony today is because this coincides with the end of a very interesting workshop organized by Jean Barbier on, it's called Youth in High Dimensions. It's a traditional event that we've been organizing for now a number of years. And the beginning of a school, of an advanced school, which is actually done in collaboration with IBM, and the organizers are Serafina Di Gioia, who is here. I don't see her, she's in the back, and also Teodoro Laino here from IBM. The school starts formally, I guess, today, but it will actually start tomorrow with the real lectures. It's a fantastic opportunity, of course, to have these two activities back to back and to have the ceremony organized precisely between the two events, also to maximize, interest in the. And the school is on more applied AI. I think it's on, if I'm not mistaken, uncertainty and robustness of DL models DL models for science. Okay, let's just wait a few minutes. Then we'll resume as soon as. Maybe I can say a bit more about IComp. Yeah, I mean, if you wish. So, I mean, Atish already mentioned this is an initiative we started already a couple of years ago at the ICTP to recognize that, theoretical physics is no longer just blackboards and pencil and paper, but it's much more than that, and that computational methods, all the way from deep learning, AI, but also data science, but also access to computational infrastructures is very relevant for whoever does research and science nowadays. And so in the spirit of what ICTP has been doing for more than 60 years now, which is to support science at the global level and especially in the global south, we decided to start this initiative that essentially is meant to provide support, opportunities for whoever is interested in acquiring these new methodologies, having more access to computational infrastructures, to do research, science, as we used to do all over the world. So the partnership with IBM is, of course, one of the most important elements of this initiative. The partnership involves not only a school on AI and the prize and the Brahmagupta prize, but it also involves a collaboration on quantum technologies, quantum information, quantum computing. So we have also a school, an essentially identical event on quantum computing, which is normally organized in spring with a prize. The prize is named not after Brahmagupta, but after Richard Feynman. The first winner of the prize was announced this year, and it's Ignacio Cirac, one of the prominent figures in the field of quantum computing. And there is also a school, an advanced school. This is also to say that whoever is here, because you're participating in the two activities, keep an eye on the ICTP program calendar of events. There's going to be much more in the areas that I presume are of interest to many of you, not only specifically on AI, but also on other aspects of computational sciences, and ICTP.IT. Still waiting for that. Maybe we can have a 5-minute break and then. Thank you, by the way. Great. David, sorry for the early time, and thanks for connecting. So we're actually a bit ahead of schedule, and so if you don't mind, we can start with your lecture. Let me just introduce you first to the audience here. The introduction to the prize is going to be given by Professor David Wishart from the University of Alberta in Canada. David got his PhD from Yale in 1991 and is currently a distinguished university professor in the departments of biological sciences and computing sciences at the University of Alberta in Canada. And his research interests are very close, of course, to those of the winner of today's prize, to this year's prize, and include metabolomics, analytical chemistry, food chemistry, natural product chemistry, molecular biology, protein chemistry, and neurosciences. Of course, with AI being a major component of his research. So, David, you have as much time as you want, actually, because we're a bit ahead of schedule, but in principle, it's 20 minutes. So, David, go ahead. The floor is yours.
Thank you very much. Hopefully, everyone can hear me okay.
Yes. Yes.
Great. I wish I could be there, but unfortunately timing didn't quite work out for me. It's a real honor to be here, or at least virtually, and to be able to introduce Mike to all of you. It's been a great pleasure working with him for the last 8 years. And what I wanted to do was maybe tell you a little bit about him, but also a little bit about the backstory to the work he's done and the work he'll be talking about today. So I think to give everyone some context, this is sort of a picture of what's inside a cell. And I think this is really important to know or understand, or maybe for people to refresh their memories about maybe some biology. Cells have DNA in a nucleus. That DNA is converted through a process called transcription to RNA. And that RNA, which is shown in red, is read through some molecular machines called ribosomes that do something called translation. And that translation converts the information in the DNA and the RNA into amino acid sequences. Those sequences, which are these little tiny balls that you see coming off from one of the blue ribosomes, have all the information needed to encode molecular machines, nanoscale machines, which we call proteins. And these self-assemble to form the kind of the purple and blue and green proteins and yellow proteins that we see in there. And those proteins perform a lot of functions, and one of the most important functions is that they both make and degrade and transport and bind small molecules. So the small molecules are the things that are shown off in the red and black. These are the tiny things that we call chemicals or metabolites or natural products. And these things can be both used for energy. Some of them are assembled and polymerized to form proteins, to form DNA, to form RNA. And it's remarkable that it's all happening in every one of your cells, every second of your life. And it's a fascinating field of, I think, information conversion, first from DNA, then to RNA, then to protein, then to structure, then on to chemicals. And this has been fascinating and fascinated biochemists for the last almost 100 years. What people have sort of determined or assessed is that there are real big problems in biology that we've been trying to solve since, as I said, the last 100 years. The first problem was, how is it that DNA is converted into proteins? DNA sequences A, T, G, and C are converted into amino acids, which are totally different chemicals and a totally different alphabet. That process of cracking the genetic code, converting DNA to codons to protein, was solved in the 60s and led to a Nobel Prize in 1968. So that was called cracking the genetic code, and it's been integral to our understanding of DNA. A few years ago, there was another problem that had to be solved, and this is, how is it that it's linear sequence of amino acids, which are these colored balls up at the top, are converted into three-dimensional structures. And those molecules, those large molecules I was showing that were purple and blue and green. And that was solved by AI. And I think many people have heard about AlphaFold, another program called Rosetta Fold. These are developed in 2021, and Nobel Prize was awarded in 2024. So we go from DNA to protein, protein to 3D structure. The other problem is, how do you go from the three-dimensional structure of proteins to what they actually make or assemble? And those are the small molecules that I was showing in the picture of the cell. And I don't know if there's a formal term for it, but I'm just calling it deciphering the chemical encoding problem, which is, how is it that you go from three-dimensional structures of proteins to produce the small molecules, or to bind the small molecules that actually do all of the work in the cell? That problem is still unsolved, and it's still quite profound. So why is that important? Why do we need to know how DNA or proteins encode small molecules? So as I've just shown, it's really the last step to solve biology. So just like the grand unified theory in physics, this is sort of the big theory or the theoretical problem in biology. The other thing that's happened is that it's much easier to sequence a gene than it is to determine the structure of a new small molecule. With today's instruments, you can sequence a gene in a fraction of a second at a fraction of a cent. And you can do an entire genome in less than an hour for perhaps 100 euros. To determine the structure of a novel small molecule can take a team of individuals up to 2 years, just for one molecule. So if we could just sequence everything and then convert sequence to all those chemicals, then our lives would be much simpler. And the reason why our lives would be much simpler is because most of the drugs that we have discovered, that we are discovering or likely discover, come from or are inspired by the small molecules found in plants, fungi, or bacteria. About 60% of all drugs today come from those molecules, from small molecules. About 90% of all drugs are small molecules. So not only do small molecules save us, but in the end, it's the small molecules that ultimately cause disease, discomfort, or death. Some numbers and statistics that people have been coming up with suggest that anywhere from 85 to 95% of mortality in the developed world comes from small molecules, effects of pollutants and pesticides, whether it's things like glucose that causes cavities or diabetes, trimethylamine oxide that causes atherosclerosis, indoxyl sulfate that causes depression. The list goes on as we find more and more small molecules that are really at the root cause of disease. What's more, that these small molecules, which we've been enumerating over more than a century, we now know that most of that chemical universe, that natural product, that metabolite universe, is still unknown. And that's because it takes so long to solve the structure of small molecules. So that's why that last problem is so important. It's also why it's so interesting. And I think this is something that probably inspired Mike when he started his undergraduate career at McMaster University back in 2011. Mike was very, very bright and an exceedingly gifted young scholar. And even before he graduated, he published a number of papers, including one in Nature Communications, that was addressing just this problem. How do we go from genes or genomes to natural products, or those small molecules? And he wrote a brilliant paper with his supervisor, Nathan McGarvey, and he actually continued to write a number of papers that appeared while he was still an undergraduate or shortly after he graduated from McMaster. These are just examples of a few. So he published in PNNS, he published in something called Nucleic Acids Research, which is a very high impact journal on programs and web servers and databases that essentially addressed at least part of this problem of how do we go from genes to chemicals, metabolites, or natural products. And he made some really interesting progress on this, and I think it certainly inspired him when he decided to go on to graduate school. So in 2015, Mike joined the University of British Columbia, and he enrolled in the MD-PhD program, the double doctor degree, which is often reserved for only the best and the brightest. And in the British Columbia program, typically you have to spend the first 2 years doing your medical training, and then in the second, or at the end of your second year, you go into your PhD program, where you have to find a supervisor, and you only have about 3.5 to 4 years to do your research, write your thesis, and then you switch back to do another 2 years of medical residency. So it's an 8-year program, very difficult. And I actually had my chance to meet Mike shortly after he started his graduate program. He had found his supervisor at the University of British Columbia, and that was Leonard Foster. And as I said, the first encounter I had with Mike was an email inviting me to join a project, an application that at the time I thought was being put together by Leonard Foster. Leonard is someone I've known for many years, and he's a specialist in protein biology and proteomics. And I was surprised to see this application from Leonard, which talked all about metabolomics. And it was a brilliantly written application. It was focused, it was easy to read, it had clear aims and objectives, the methodology was spot on. And I remember writing and even, I think, talking to Leonard and saying, Leonard, I didn't know you were so into metabolomics. You've written just a brilliant proposal. And that's when Leonard said, I didn't write a word of it. It was all Mike. And I think that's when it dawned on me that we were dealing with someone who was quite special. So here's someone in their very first year in graduate studies who was writing a grant proposal for a large project in computational biology, who had done all of the research, come up with the, I think, fascinating ideas that are sort of pictured here, and had done all of the background research. And I think that happens maybe only once or twice in a person's academic career, where you encounter a graduate student who's far better than you. And that's when you have to decide to sort of put your ego in check and let them lead the way. So the components of this grant probably are too small to read, but many of these things form the basis to the work that Mike is still doing today, and which led to a number of very important discoveries, for which he's been awarded this prize today. All of those nascent concepts are actually in this picture, or several of the pictures. And in fact, Mike has sort of continued to lead in that process. The title of his project was called Illuminating the Dark Matter of the Metabolome with Convolutional Neural Networks. So maybe I'll explain a few things. The metabolome is sort of the collection of all of the metabolites that we find in cells. The proteome is the collection of all the proteins, and the genome is the collection of all the genes. So in mammals, like humans, there's about 20,000 genes. Those code for about 150,000 proteins. And those proteins act, synthesize, degrade about 250,000 chemicals that we know about. Those are called the metabolites. So the study of the metabolome is metabolomics. The study of the proteome is proteomics. The study of the genome is genomics. With technology today, we have tools, methods, techniques, instruments that can basically cover all the genes in the genome on any organism, pretty much all the proteins, because we could know the genetic code. But in the case of the metabolome, we only have about 5% coverage, and that's a problem. And that's a problem that Mike recognized. And so that dark area is that what we call the dark matter. Now, the reason why Mike had contacted me was that for many years, I have been working on a project called the Human Metabolome Project. So that was sort of the equivalent to the Human Genome Project, which did the DNA genome of humans and many other organisms. There's another one called the Human Proteome Project, where they're characterizing all of the proteins in systems. And then about 20 years ago, we had started a Human Metabolome Project. We were developing both the tools and the databases and the infrastructure to do metabolomics. Not at the same scale as the proteome or genome projects, but Mike was aware of the work we were doing, and that's why he reached out to me to work with him on this project of illuminating the dark matter of the metabolome. Now, in metabolomics, what we do is we take samples and we grind them up, we extract them into fluids, and then we inject them into things like mass spectrometers and NMR spectrometers. And that's something that people have been doing for more than 100 years, actually, whether it's through mass spec, chromatography, now nuclear magnetic resonance. It's this last step in this pipeline where we do the data analysis, which is where things are sort of falling down and where most of the work and most of my work has been to try and figure out these messy spectra, what's in them, what they correspond to. And I think, as I'd said, we haven't, even with 20 some years of working at this, we haven't progressed very far. That's the dark matter problem. So most of the compounds that we know or think are in the metabolome are not known or not yet identified. That's not like it is in the case of genes or proteins. When we look at our mass spectrometer signals and our NMR signals, but particularly with the mass spectrometers, most of those features or peaks we can't identify. They don't match to anything in any of our databases. They don't match to anything we've been able to isolate or find. As I said, it takes up to 2 years with a team of people to identify a single compound. Right now in metabolomics, the best we can do is maybe identify about 2,000 compounds at a time. That's only less than 1% of the known metabolome, and it's less than 0.1% of what we believe is out there. So all of these unknowns, the ones we can't identify, the ones we haven't identified, the ones that we think are out there, that's all part of the dark matter of the metabolome, and that's a mystery. So Mike's solution, both in that proposal or as it evolved over the course of the few years we worked together on it, was to use something called chemical language models, or CLMs. So I think most people are familiar with large language models, and those are the ones that make up ChatGPT or Claude or everything else. And the idea is to take lots of text, lots of examples of English text, lots of words, apply deep learning, and then you have a tool that can put those words into meaningful sentences. Mike's idea was to extend that to chemicals. And it turns out that in the 1960s, people developed a chemical language called SMILES. And so you can write a chemical in text rather than drawing it out. And so you can give your deep learning model, something like a long short-term memory, deep neural net, or convolutional neural net of some form, or transformer, and it will learn the patterns of those characters. And not only will it know or learn those ones that it's been fed to, but it'll start creating and put things together to make new words, new chemical words, and therefore new structures. So we can convert those text strings into chemical structures, which is how most chemists think. So that's the essence of a chemical language model, to take text, run it through deep learning, and generate new text, which corresponds to new structures, or novel structures. Now that's maybe just an interesting parlor trick, but Mike found out some very interesting things along the way and made some really important innovations. So as I said, you take some known structures, you convert them to text strings called SMILES. What Mike realized is that you can do something with this equivalent of synonyms. So just like in languages, there's same spelling for same words, or same words, or different words can be used to mean the same thing. So this is called enumerated SMILES. It's a form of data augmentation that Mike realized we could do. So the same SMILES or different SMILES strings mean exactly the same structure. And you can generate hundreds of SMILES strings for the same structure, which are like synonyms. So by augmenting sort of minimal data sets, he was able to build a very large training set and therefore train these deep learning models with the amount of data they really need. And so he ran them through LSTMs, and he produced all kinds of, not only the structures that he trained on, but a whole bunch of novel ones. And what Mike also realized was that the frequency at which these novel structures were being generated actually had some information in them. So the most frequently generated structures, it turned out, were the ones that were most likely to be synthesized. These are the ones which were chemically or biochemically compatible with the known rules of chemistry, the known rules or still to be discovered rules about isomerism and ortho, meta, para substitutions. So these CLMs learn chemistry, not just characters, and they learn biochemistry, not just simply chemical reactions. And so by using this frequency or prior probability, Mike was able to realize that you could use these models to create collections of novel, biochemically feasible compounds and assign a probability to their existence. And that's really quite revolutionary and transformative. So with that idea, Mike and Leonard and myself and our team started working on this idea called in silico metabolomics. That's largely formulated in Mike's grant, where the idea was to take the known compounds that we had compiled through the Human Metabolome Project, use AI to generate novel compounds, so going from 250,000 to on the order of 3 million novel compounds, and then also to use AI to convert those structures into observables, things that we can see like mass spectra or NMR spectra or attention times, things that we measure. So then we can map what we can measure to actual structures. And so it becomes kind of a library lookup. So that led to a program or set of tools that Mike had developed along with us over the last 4 or 5 years. The first one was to develop programs to identify novel psychoactive substances, but then we started applying it to the original project, which was the metabolome. And that led to a program called DeepMet, with the intent to identify molecules in cells or in tissues or in biofluids, and to identify those novel compounds, those dark matter that we couldn't identify. And that actually led not only to this paper, but many other papers over the last 8 years that I've had a chance to collaborate with Mike, or more, I guess, a chance to ride along in his wake. This was published in Nature earlier this year, which described DeepMet and the ability to use the chemical language models and this in silico metabolomics approach to identify novel compounds. And as I said, it can take up to 2 years, but with this paper, we showed that it was possible to identify compounds in literally a matter of minutes. Although to get it published, we had to synthesize all the compounds or buy them, which took a much longer time. But the concept was proved with this, and I think it's a very exciting result, and there are many other exciting results that I think Mike will explain later. But this is the culmination of that grant and that collaboration, and all of it, I think, is really due to Mike's insight and brilliance. So I talked about the 3 big problems, and I think over the last 10, 11 years that Mike has been an active scientist, he has done probably more than anyone else to be able to go from this idea of encoding the genes to the chemicals. You can see it from his early work as an undergrad to his work as a PhD student, and now to his work as an assistant professor at Princeton. So with that, I wish I could be there to congratulate Mike, but I also wanted to be able to give you a bit of a backstory about the amazing things that Mike has done and the amazing person that Mike is. Thank you.
Thank you very much, David, for this wonderful and very clear, insightful introduction to the work. We all now can't wait to hear from Mike what he's done. So I will now like to call Professor Michael Skinnider from Princeton University to the podium for his talk. The title is Charting the Unknown Metabolome with Biochemical AI. Mike, the floor is yours.
Okay, well, I want to start with some gratitude. It is really, I can't tell you what a pleasure it is to be here speaking to you today. And so I have a lot of, people to thank. First of all, the selection committee, I can't say enough how meaningful it is to me to be accepting this award from ICTP and IBM. The organizers of this session, and David, thank you for this very, very far too kind and quite touching introduction. I'm going to spend a little bit of time telling you about my group's work, as David alluded to develop biochemical AI models that allow us to predict the existence of undiscovered metabolites. And I'll tell you about how we've actually used these models to make what I think are quite interesting experimental discoveries. I should preface this by saying it's a little strange, even a little intimidating to be speaking to this audience because, as you can probably tell from David's introduction, I'm certainly no physicist, not even a theorist. We work on very applied AI for chemistry and its intersection with biology. And so I certainly, was not, trained in physics. As David mentioned, I was trained in medicine and computational biology, and before that, of course, I was an undergraduate student at McMaster University, and this is really where I developed a deep interest in metabolism and the technologies that we have available to us to measure metabolism. And so in Nathan McGarvey's lab, the focus was on bacterial metabolism. Bacterial metabolites are, as David mentioned, the basis for many of the most important drugs that we use in the clinic today. Many, critical antibiotics and anti-cancer drugs. And so my work in Nathan's lab had developed machine learning models to help discover these bacterial metabolites faster using their genome sequences and then also their metabolomes. This work ultimately was spun off into a company that I worked for several years. But when I moved to the west coast of Canada to earn my MD and my PhD, I moved into a different field, one that also uses mass spectrometry. This is the field of proteomics. And I worked with Leonard Foster on mapping networks of protein-protein interactions, which sort of constitute the architecture of the proteome using a technique called cofractionation mass spectrometry. And so this was my thesis itself. I also spent some time in Lausanne as a visiting PhD student with Grégoire Courtine's group. And my work there was focused actually on neuroscience, so I developed machine learning methods to identify specific subpopulations of neurons that are involved in the production of behaviors such as walking. And this was really exciting work. It culminated in the discovery of a subtype of neuron in the spinal cord which can be electrically stimulated in order to reverse paralysis and restore the ability to walk, both in mice and in human patients. So these are obviously three very different areas, right? I told you about using genomic data, using transcriptomic data, using proteomic data. And those are very disparate fields of biology. But I think they have one thing in common, which is that in all three of these fields, the technologies for measuring these molecules, these biomolecules, are very mature. No matter where you are in the world, there will be a core facility somewhere down the road who can take your sample, whether that's a tumor biopsy or a grain of dirt or a plant leaf, and they can measure essentially all of the DNA, RNA, or protein in that sample. And this is just not the case for small molecules. Today, it is still essentially impossible to measure all the small molecules in a given biological sample. It would take, a team of many dozens of people, many years, and a large investment of money. So to me, this is quite a profound gap in our ability to measure the natural world and therefore understand the natural world. It's also a very exciting problem as a computational biologist because I don't think it's for lack of having the right technique to measure these small molecules or metabolites. I'm going to use these words interchangeably, but a metabolite is just any small molecule that's produced by a living organism. So we already have an instrument that can measure all of the small molecules in a given sample, and that instrument is called a mass spectrometer. A mass spectrometer can detect thousands of small molecules in minute amounts of biological material, and not only does it detect these molecules, it collects very rich data for every one of them. And so maybe you can see how this starts to seem like a computational problem. How can we take this rich data that we're getting from the mass spectrometer and deduce from this data the chemical structure of the small molecule that was present in our sample? And that, again, as David foreshadowed, turns out to be very difficult. And today, when people measure biological samples with mass spectrometry, only a very small fraction of the small molecules that they detect can actually be identified, that is to say, linked to their chemical structures. So people realize that there's this discrepancy between the number of small molecules we can detect and the number we can identify, and this has led to a lot of speculation that perhaps these unidentified molecules are actually unidentified because they're unknown to humanity. And that speculation has been borne out over the last decade. Many new small molecules have been discovered that have been shown to play important roles in human health and disease. I'll talk just about 3 examples to sort of crystallize why this is an exciting and important problem. The small molecule at the far left, this molecule called colibactin, is produced by E. coli that live in some of our guts, not all of our guts. And this molecule binds to DNA, damages that DNA, and in fact directly causes colorectal cancer. So this is a very important molecule for human health. The one in the middle, lactose phenylalanine, is also an important molecule for human health. This is a molecule produced not by the bacteria that live in us, but by our cells themselves. And this molecule acts to suppress appetite, and its levels are linked to a person's risk of developing obesity, and with obesity, one of the 13 cancer types that is caused by obesity in part. And then the molecule at the far right is produced by human cells when a patient is treated with a synthetic drug that's used to treat pancreatic cancer. And when this chemotherapeutic drug is metabolized by human cells, it is no longer active. So depending on how much of this metabolite is produced, a person's cancer treatment may or may not work. So hopefully you can see that these are, important molecules to know about. Their discovery has not been easy. Each one of these projects, each one of these discoveries required years of work. And that's because historically discovering new metabolites has been very slow. As David said, it can take years. We knew about colibactin for more than a decade before it was actually, the structure was actually solved. So putting this all together, this is why we're so excited about the prospect of developing biochemical AI models that can allow us to decipher this so-called chemical dark matter and ultimately to chart the complete map of the human metabolome in analogy to the Human Genome Project and its success almost 25 years ago now. So this is sort of a grand challenge. People have been interested in deciphering mass spectrometry data for many decades, going back to the 1960s. You might ask, how are we going to make a dent in this fundamental problem? And I'll talk about one approach that we've had a lot of success with, which is based on sort of the following premise. Clearly, there's much to learn about the metabolome, but on the other hand, we do already know a lot. We've been doing biochemistry for 100 years, and we've come to understand a great deal about human metabolites and the enzymes that make these metabolites. So can we now leverage this information, this wealth of information that we have about known metabolism, in order to predict the structures of metabolites that are likely to be discovered in the future? As David set up nicely, the technology that we've been using to do this is based on what are called chemical language models. So the idea here is that we can represent the two-dimensional molecular graph of a metabolite as a one-dimensional string. We are particularly fond of this format called SMILES, and I'm showing you here how a SMILE string allows us to express a graph in text. And of course, we do this because representing metabolites as text allows us to use the same kinds of neural networks that have been very successful at learning the syntax and semantics of human language. And rather than applying those models to the words in a sentence, we can effectively apply them to model the atoms and bonds in chemical structures. So concretely, our approach uses an autoregressive language model. Much of our work so far has used long short-term memory networks to model a sequence of characters in a SMILE string. We train this model by minimizing the cross-entropy loss of next token prediction. So at each position in this sequence, we ask the model to predict a probability distribution over potential characters at the next position in the sequence, given the sequence of characters that's come before. And then once we've trained this model, we can sample unconditionally from it in order to generate new SMILE strings and therefore new metabolites. At the outset of this application to metabolism, we had some proof of concept for the general idea that this approach could be successful, as David also alluded to, in the setting of forensic chemistry, and specifically predicting future illicit drugs of abuse. So the structures of metabolites kind of reflect the enzymatic logic of biosynthesis. In contrast, the structures of illicit drugs reflect the human logic of medicinal chemistry. These drugs tend to be made from a small number of scaffolds or starting points, and those scaffolds are derivatized or transformed using a fairly small vocabulary of chemical transformations. And I think that you can probably appreciate this from the series of fentanyl derivatives that I'm showing here. You know, even if you're not a chemist, you can probably see that these structures all have something in common. And so this idea had motivated us to train a language model on the structures of all known illicit drugs of abuse. This is a model we named DARK-NPS. And we did something that in the field of AI and machine learning has become a little bit unusual after we trained this model, which is that we did nothing at all. So we waited for 6 months after training this model. And over that 6-month period, many new drugs were being discovered by forensic labs around the world. And at the end of this prospective observation period, we asked how many of these newly discovered illicit drugs did our model correctly anticipate. And we were quite excited to see that our language model correctly generated more than 90% of these newly discovered designer drugs, as they're often called. I'm showing you some examples here of novel psychedelics, novel opioids, and novel stimulants that our language model correctly generated. We then went on to show that we could combine this language model with mass spectrometry data, and what this allowed us to do was to take a seized drug product or even a patient sample and perform structure elucidation, that is to say discovery of previously unknown illicit drugs with quite a remarkable level of accuracy. And in fact, we worked with the Danish National Forensic Lab to help discover a new illicit drug, this derivative of the street drug PCP that I'm showing you here. So I'm going through this work fairly superficially, but all just to make the point that we had some basis for optimism that a very similar approach would, instead of learning the medicinal chemistry logic of illicit drug synthesis, allow us to learn the enzymatic logic that gives rise to the structures of metabolites and predict the metabolites likely to be discovered in the future. So this is exactly what we did. We trained a chemical language model that we called DeepMet on the structures of all known human metabolites. This is work that was led by my PhD student, Tony, and I'll tell you about some of the things we found DeepMet can do. So I mentioned a moment ago, our hope was that this model would sort of come to learn the logic that gives rise to the structures of known metabolites and then be able to generate new structures that could potentially be metabolites. And we were able to show that the structures that DeepMet generates do indeed look like they could be metabolites. We did this through several lines of evidence. The first of which is shown here. It's just by visualizing the chemical space of the known metabolites in the training set and the generated molecules. So in these plots, the X and Y axes are showing chemical space in 2 dimensions. The known metabolites are shown in color on the left. The generated structures from the language model are shown at the right. And hopefully you can appreciate that there's relatively few points or structures that are exclusively occupying certain areas of chemical space. And so, at least qualitatively, this suggested to us that our model was generating metabolite-like structures. Of course, we also wanted to evaluate the model more quantitatively. We did that with a series of analyses. One that I'm particularly fond of is we asked whether we could train a second machine learning model to classify whether any given structure was a known metabolite from the training set or a generated structure from the language model. We found this classifier, in fact, could not really tell apart these two categories of structures with an accuracy all that much better than random guessing, so the implication being the generated structures are almost indistinguishable from the known metabolites. One thing we noticed over the course of these analyses, again, as David sort of set up, is that these structures the language model generated were not all equally likely to be generated. So this plot is showing you the distribution of frequencies with which every unique chemical structure appeared in a sample of 1 billion SMILE strings that we drew from our trained language model. At the top left, you can see there are millions of structures that are generated once or twice or a handful of times. But at the bottom right is a long tail of novel structures that are not in our training set of known metabolites, which are generated thousands or tens of thousands of times. So we thought that maybe the frequency with which any particular structure is being generated by our language model, no matter what smiles it uses to write out that structure, could be viewed as a Monte Carlo density estimation of the probability of that structure given the parameters learned by the model from the training set. And it was therefore very interesting to us to understand what are these frequently generated structures and why are they viewed as such probable extensions of the training set. And we noticed that these frequently generated structures had a number of interesting properties. So for one thing, the more often a structure was generated, the higher degree of structural similarity it tended to show to a known metabolite in the training set, suggesting that the frequently generated structures are more metabolite-like. And not only were they more similar to known metabolites, they were also more likely to be known metabolites from a held-out set that we had deliberately withheld from our model when we trained it. And so this finding in particular got us thinking that perhaps DeepMed had developed some ability to fill in the gaps in our understanding of mammalian metabolism. So to test that idea more thoroughly, we did an experiment very similar to the one I just described for dark NPS. We located a set of 313 metabolites that were discovered after we finalized the parameters of our model, and we asked how many of these newly discovered metabolites did our language model correctly generate. We were very pleased to see that the model successfully generated the vast majority of these newly discovered metabolites, especially once we removed those that were not really endogenous mammalian metabolites, but which were instead metabolites of prescription drugs or synthetic pollutants or diet-derived compounds. And our model not only generated these newly discovered metabolites, it generated them frequently, so frequently that we could in fact separate this background of all generated structures from these small number of newly discovered metabolites with a remarkably high degree of accuracy. The implication being that with this model, we can quantitatively prioritize the structures likely to be discovered in the future. So the data that I've shown and some other experiments we had done to this point suggested to us that we might have an ability to predict the structures of metabolites that are yet to be discovered. And we wanted to put those predictions to the test experimentally. We did this working with Aaron Shapiro, a scientist at the BC Centre for Disease Control in Vancouver. What we did is that we synthesized or bought chemical standards for a few dozen of these frequently generated structures. We ran those pure compounds on a mass spectrometer, and we compared the data from these chemical standards to mass spectrometry data from thousands of urine and blood samples that Aaron and his lab had analyzed over the course of their routine operations. And through these experiments, we were able to discover 17 previously unknown human metabolites. I'm showing you two examples here. At the left is a chemical structure. At the right is the mass spec data from the pure synthetic compound and the human urine. And hopefully, you don't need to be an analytical chemist to appreciate that the top and bottom halves of these plots look virtually identical, indicating that we assigned the correct structure. So given that it can take months or years to discover even a single new metabolite, we were quite excited that we were able to discover 17 just by predicting that they ought to exist. This was, as I said, exciting, but it came with an important caveat, which is that this is not typically how metabolite discovery is done. Usually what happens is you collect mass spectrometry data from two groups of samples. Let's say, for instance, tissues from a person with a disease and tissues from healthy control individuals. And you then compare the abundance of the signals in these samples and find that some unknown signals are up or down regulated in the diseased patients. And you want to discover those specific signals that were detected by mass spec and are displaying some interesting response to a perturbation. So we wanted to integrate our model more directly with mass spectrometry data. I'll tell you about a few different ways we did that, but I'll start with the simplest, which is that we thought we could simply filter this large database of generated structures on the basis of a single experimentally measured property, such as an exact mass. And I should highlight that this is one of the simplest and least ambiguous properties to measure experimentally. We can quite readily measure the masses of all the compounds in a given sample. So I'll show you an example of what this process looks like given as input an exact mass of 176.0950 Daltons and a small window of error around that mass. And all the structures that I'm about to show you were deliberately hidden from the language model. So this is simulating the discovery of an unknown metabolite with this exact mass. So DeepMed generates actually several thousand structures that could conceivably explain this exact mass, but they're not all equally likely to be generated. In fact, as you can see from this plot, which is showing the relative frequencies with which the 20 most frequently generated structures appeared in this sample from the model, one structure is assigned a much higher probability than any other. That structure turns out to be the one I'm showing you here. This is 5-hydroxytryptamine, also known as serotonin. This is a very well-studied, perhaps even a famous metabolite. It's a neurotransmitter. It's targeted by many antidepressant drugs. The second most frequently generated structure with this mass is also a known metabolite. This is cotinine, which is the major human metabolite of nicotine. So this is a metabolite that insurance companies around the world test for every day. And many of the other top-ranked structures are sort of variations on these themes. So the key point here is that given really minimal data, as little information as a mass, our model is capable of producing a ranked list of hypotheses about the complete chemical structure of an unknown metabolite, and to our knowledge, it is the first and still only tool that can do this. You might well ask if this example is a bit cherry-picked, and in reality, we did the same analysis with every known metabolite in our training set, withholding it from the training set, simulating metabolite discovery, asking how well we could recover its exact chemical structure from its mass alone. We found that our model was surprisingly accurate. In fact, it could prioritize the correct structure about 30% of the time and rank the correct structure in its top 3 predictions a little over 50% of the time. This is pretty exciting because this is a field in which accuracy historically has been measured in single digits. But it also comes with a caveat, which is that mass alone does not actually differentiate any of these metabolites I just showed you. Knowing the mass of a molecule does not tell us whether it's, for example, serotonin or cotinine. And so most metabolomic studies rely heavily on a technique called tandem mass spectrometry, or MSMS, which basically involves taking a molecule, colliding it with an inert gas, which fragments the molecule into a series of pieces, and then measuring the masses of these fragments. So it was a very logical next step for us to ask, can we integrate our language model with MSMS? We did this in collaboration with David's group and his PhD student, Fei Wang in particular. Fei developed a series of different machine learning models that all have in common that they take as input a chemical structure represented as a graph and predict as output the MSMS spectrum of that structure. And we can compare the predicted and experimental MSMS spectra, formulate some score that indicates the quality of the match, and then this score can be viewed sort of loosely as proportional to the likelihood of the observed spectrum given the structure. We can combine this with our language model, which gives us sort of a prior over possible metabolite structures, and get this combined score that in practice improves considerably on the performance of the language model alone. We can now solve the structures of unknown metabolites about 50% of the time and rank the correct structure in the top 3 around 2/3 of the time. And without going too much into the weeds of mass spectrometry, MSMS isn't the only thing we can measure. We can measure a number of other properties in a mass spectrometer. We develop machine learning models to predict each of these properties. We combine those predictions with the prior from our language model using a second layer of machine learning. This is a technique known as meta-learning. And what we get out of this process is a score that quantifies the match between a given hypothetical structure and the totality of the analytical data for any signal detected in the mass spectrometer. This approach further improves the accuracy. We can now solve unknown metabolite structures with an accuracy of about 70%. Again, this is a problem on which accuracy historically has been in the single digits, so this is really quite an impressive level of success for this field. And so once again, all this data suggested we had quite a powerful method on our hands. We wanted to put it to the test experimentally and see, can we use this approach to actually make new biochemical discoveries? We did this in collaboration with Josh Rabinowitz, my colleague at Princeton, who's an expert on metabolism, and his group had just profiled 23 mouse tissues by mass spectrometry. And so we used DeepMet to assign chemical structures to many of the signals in this data set, and again, we went and synthesized a series of the predicted structures to see whether or not our model got them right. And through these experiments, we were able to discover another 16 novel mouse metabolites, each confirmed by synthesis. I'm showing you just one example here. The structure assigned by our model is at the top left. At the bottom left is the abundance of this particular metabolite across tissues, and you can see that this molecule is exquisitely specific to the mouse small intestine. And so we think that the reason this molecule went undiscovered until now is because the small intestine is just a relatively uncommon tissue to go looking at for metabolite profiling. Other metabolites are more ubiquitous. This is an example of a molecule that is present at low levels in many different mouse tissues, and perhaps this one has gone undiscovered just because it's relatively low in abundance. I should say that both of these molecules are new metabolites. They were not known to be produced by living organisms, but they were known in a more abstract sense that they had been made by humans in the lab synthetically and were present in existing databases of chemical structures. And so, in principle, though perhaps not in practice, they could have been found with existing approaches. But one thing that's quite unique about our approach is that it gives us the ability to discover metabolites that are not present in any database of existing chemical structures, are not even known to humanity at all. And this particular metabolite is exemplifying that possibility. This is a molecule called 2-sulfoglycerate that was not present in any existing database, but which we were able to discover nonetheless. So we've been able to scale this approach up now. We've now used DeepMet to discover more than 90 novel metabolites. I think this is really exciting to me because the biochemical discovery is really the driver of this work, and I think it highlights how this model and the ecosystem of models we've developed around it can meaningfully help us complete a map of the metabolome for the first time. So it's worth pausing to sort of think about, why is this approach as successful as it is? And I think a very appealing explanation is that these chemical language models effectively allow us to narrow the search space when we know we're hunting for a particular kind of small molecule. Chemical space as a whole is sort of incomprehensibly vast, but metabolite-like chemical space is a very small subset of that vast chemical space, and by using the language model to generate only metabolite-like structures, we can effectively make this problem much easier for ourselves. But with that being said, this approach is far from perfect. I don't want to give the impression that we've solved this problem. Still, in the lab, we find that when we synthesize a predicted structure, a little over half the time it's still incorrect. And this is, on the one hand, quite an impressive level of performance for a tool in this field, but on the other hand, it's not a sufficiently high level of accuracy to really comprehensively master the metabolome at scale. And so the implication is we need to do one of two things. We need to make better use of the data we already have, or we need to acquire new data and develop bespoke models for that new data if we want to improve the accuracy and throughput of these approaches. So we have been thinking quite a bit about how to make better use of the data we already have, starting with MSMS spectra. So my postdoc Vishu has recently described our efforts to comprehensively assemble all of the MSMS spectra from small molecules in existence in a database he calls Spectraverse, which is about 3 times larger than any other collection. Vishu did this, of course, because he wanted to start training new, better AI models to decipher MSMS spectra. But rather quickly after he started doing that, he ran into or discovered what we believe to be a pretty serious problem with the way that these models historically have been evaluated. Because Vishu finds that on existing benchmarks for interpretation of MSMS spectra, we can match or even exceed state-of-the-art performance using a trivial model that never actually sees an MSMS spectrum at all. And so it turns out that, to make a long story short, the kinds of molecules that have been measured by MSMS in the past look very different from the space of all possible molecules found in chemical databases. If you show the two molecules at the right to an analytical chemist or a natural products chemist, or really any chemist, they will sort of know instinctively the one on the top looks like something that could occur in a living system, but the one on the bottom looks like a synthetic molecule that could only be made in the lab. And so models can cheat, and instead of learning anything about the physics of MSMS or the behavior of small molecules in the mass spectrometer, they can just learn to recognize what molecules look like they might have been measured by MSMS in the past. And when we encourage models to cheat in this way, again, we can achieve state-of-the-art performance. So we think that this is a pretty serious confounding factor. It might explain why there's been relatively little progress on this problem over the last 10 years. Perhaps people have been optimizing their models against what is essentially a spurious signal. I'm not going to talk in too much depth about that. I want to spend just the last few minutes here telling you about another project in which we asked ourselves, can we, working with experimentalists, go out and experimentally acquire the exact kind of data we wish we had to train an AI model on? And so this is a project, again, that was a deep collaboration with Josh Rabinowitz. It was jointly led by Justine in my group and Mike MacArthur and Wenyun Liu in Josh's group. So the data we wish we had to train AI models for metabolite discovery, in the end, is data produced by a technique called isotope tracing. This is a really long-standing and powerful technique. The basic idea is to treat cells or tissues with an isotopically labeled form of a metabolite. So, for example, if you take glucose, in which every carbon-12 atom has been replaced by carbon-13, and you give that 13 carbon labeled glucose to cells, you will see a signal appear in the mass spectrometer where lactate should be, but with a mass about 3 Daltons greater than lactate. And this is because that 13 carbon labeled glucose is being metabolized into 13 carbon labeled lactate. So this is a technique that's been used historically with great success, but in a very quantitative way. It's been used to map or measure flux through a small number of well-studied metabolic pathways into a handful of central metabolites, and more often than not to develop quantitative models of metabolic activity. And we sort of developed a more ambitious, or at least large-scale vision. We asked, could we perform many isotope tracing experiments in mice and measure the metabolomes of many different tissues, and thereby track all of the downstream products that these isotopically labeled precursors are used to make? And in doing so, we reasoned that for any given unknown metabolite, we could tell what that unknown's precursors actually are. So this is exactly what Josh's lab did. They took 26 different isotopically labeled metabolites, they treated mice with these metabolites, and they collected mass spectrometry data from about 20 different mouse tissues. And Justine then developed a statistical approach that takes this very large and complex data set and reduces it to a series of statistically significant labeling events. So what Justine gets out of her model for each peak, whether known or unknown, is a matrix of p-values in which the columns are precursors and the rows denote the number of carbon atoms incorporated from each of those precursors. And so in this matrix, you might be able to see that the only statistically significant labeling event is the incorporation of 6 carbons from tryptophan. It turns out that this particular molecule is a known metabolite, niacinamide or nicotinamide, which is known to be made from tryptophan. So this sort of validates the performance of our approach, and hopefully you can imagine that this would be very useful to know if we did not, in fact, know the structure of this particular molecule. Justine then showed that we could integrate these labeling patterns with, again, DeepMet using a technique known as cross-modal contrastive learning. So this has been an extremely influential paradigm. I'm sure most people here are familiar with it, but in case you're not, still sort of the best known application is aligning learned representations of images and their captions. So essentially one trains 2 separate neural networks to encode images and their captions, and then trains those neural networks so as to maximize the similarity of the learned representations for image caption pairs while simultaneously minimizing the similarity of learned representations for unpaired images and captions. And it turns out that we can do essentially the same thing, but instead of images and their captions, we can align learned representations of labeling patterns and chemical structures. And so what we get out of this is a latent space in which we've jointly embedded hypothetical structures from our language model and the labeling patterns measured by mass spectrometry for unknown metabolites. And we can then rank the hypothetical structures based on their consistency with the observed labeling pattern, given everything that we know about known metabolism. We've been able to use this approach to discover really some structurally very intriguing metabolites. I'll show you just one. This is a metabolite that our statistical model tells us incorporates 3 carbons from cysteine, 2 carbons from methionine, and 2 carbons from various precursors to acetyl-CoA, such as sugars, acetate, or fatty acids. Our model assigned the structure at the top right. We synthesized the structure and confirmed that it is in fact correct. And this was quite an exciting structure to us because the functional group that's attached to the cysteine sulfur atom, this dithioacetal, has actually never been described in the context of mammalian metabolism. It's known in a handful of bacterial metabolites, but it was not known to be made by mammals. So this is quite structurally unprecedented. And we found that, in fact, a series of other metabolites also share this functional group, all of these, again, being confirmed by chemical synthesis. So this is quite a unexpected discovery enabled by this model. I'm going to skip over some of the other, structurally very intriguing metabolites that we discovered with this model and just tell you about some biologically intriguing metabolites. So to sort of exemplify the utility of this model to better understand health and disease, we went looking for metabolites that are changing significantly in age. We analyzed a data set that Josh and his group had published a year prior, where they looked at a large cohort of young mice and followed them as they aged. And we set our sights on the 2 most significantly age-responsive metabolites in the mouse metabolome, both of which were unknown, and we were able to solve both of their structures. The first one is actually the metabolite I just showed you, this dithioacetal-bearing cysteine derivative. The second metabolite, which is perhaps less structurally interesting, but even more biologically interesting, is the one at the bottom left. So this is a 15-carbon unsaturated fatty acid that's made from a precursor called mevalonate. Mevalonate is the same precursor to cholesterol, of course, a very well-studied metabolite relevant to health. And so we named this molecule 2,3-dihydrofarnesoic acid. Both of these molecules are profoundly dysregulated in age. In every mouse tissue we looked at, they are both downregulated by at least 10-fold, sometimes considerably more than that. We couldn't actually identify the cysteine derivative in our available human samples, but we found that dihydrofarnesoic acid is also markedly downregulated in aging humans. So you can see from this plot, it's the levels of dihydrofarnesoic acid in the blood are about 5-fold lower in individuals 50 or older than they are in individuals 20 or younger. To make a very long and complicated and ingenious series of biochemical experiments short, we were able to learn quite a bit about the function of this molecule. All the credit for these experiments goes to Mike MacArthur and Josh's group. It turns out that, like I said, this molecule is made from the same pathway that makes cholesterol, and Mike discovered why exactly it's downregulated in age. It has to do with a specific enzyme called pantothenate kinase, which is involved in the conversion of a vitamin called pantothenate or vitamin B5 to an important precursor called acetyl-CoA. And so Mike found that this enzyme is dysregulated in old mice, and that leads to a defect in CoA synthesis, which leads to a decrease in dihydrofarnesoic acid. This enzyme is targetable pharmacologically by existing drugs, and it turns out that you can take one of these drugs, give it to mice, and this restores synthesis of CoA and production of this dihydrofarnesoic acid in the aged mice to levels closer to middle-aged, or in some cases even young mice. And Mike and Josh are continuing to study the myriad beneficial health effects that doing so appears to have for aged mice. So I think, without going on a long tangent about those health effects, I think this exemplifies the promise that I sort of opened with about how understanding this chemical dark matter of the metabolome could help us discover new metabolic pathways, understand how those pathways are dysregulated in health and disease, and ultimately lead to new therapies. So I'll end there. if you leave with anything, I know this might have been a bit outside of the wheelhouse of this institution or this workshop, but I hope I left you with some appreciation for the idea that there's a vast chemical world within our own bodies and in the world around us that we simply do not have the ability to access or measure at this point. And I think that that is not the fault of existing analytical tools. It's a computational bottleneck, and it's one that I think AI can meaningfully solve. I've shown you some of our work on doing just this. Our chemical language model DeepMet that I showed can predict the metabolites likely to be discovered in the future, and which we've interfaced with mass spectrometry data to enable automated metabolite discovery. And then I talked a bit about how we further integrated DeepMet with this systematic isotope tracing data and how this allowed us to discover some chemically and biologically fascinating metabolites. The work I showed was led in my group by Tony, Vishu, and Justine. Both projects were collaborations with Josh Rabinowitz, and the tracing project in particular, again, was led by Mike and Wen-Yoon in his group. And then, of course, the DeepMet project was a collaboration with David and Aaron Shapiro. And really, I have to thank a very large group of people who made many important contributions to all of this work. Of course, also thank the funding sources that allowed us to do the work. And last, you for your attention. Thank you very much. Okay, sure, yeah.
We're having some questions since we are a bit ahead of time. So thank you very much. First of all, it's really beautiful. Thanks for making an effort, actually, to make it understandable to a broader audience. Is there any question from the audience? Okay.
So thank you very much for this very nice talk, understandable even to people like me who are not from biology, so thank you very much. So I like particularly this idea of using kind of chemical structures, translating into smiles, and then using text-based AI in kind of to discover new molecules. So we all use things like ChatGPT and language models for chatting. And one of the big issues there is that sometimes the AI hallucinates and just tells us things which are not true. And the same surely also happens in your models, that you get out structures which are simply not true. So did I understand well that kind of your filter to find out when something is true or not is the frequency with which a given structure is suggested, or are there other tools?
That's an important component, absolutely. I guess what I would maybe add to that is ultimately the analytical data is our filter. In many of these approaches are kind of inspired by or borrow from analogous approaches that are used in drug discovery, but in drug discovery, it's, you know, figuring out whether the molecule of interest actually has the activity that you hope it does is a hard thing to estimate in silico, whereas for us it's much easier to kind of have some sort of approximate metric about how well does this generated structure match the mass spectrometry data. And so we can, I think maybe it would be a bigger percentage of the filtering is done by checking this database of generated structures against the actual analytical data that we acquired from, you know, tissues or biofluids to see whether such a molecule might exist. I hope that answers the question.
So I just have a curiosity. Is it possible to refine the search in such a way that you look for unknown molecules with certain properties or functionalities, or once you find out, it's easy to determine those?
Well, no, far from it. It's not easy to determine the functions of these unknown molecules unless we have some sort of strong prior reason to go after them in the first place. In the aging data set, of course, we knew that these molecules were changing with age, and so that gave us some ideas about what they might be doing based on what's known about the biology of aging. And so that's kind of, I mean, this is another major unsolved problem, is once you know the structure, what is it doing? I think what is it doing, of course, is a very nebulous question. Function is not kind of a well-defined problem, but I think an important component of that is what proteins does the molecule interact with? What enzymes are making it or degrading it? Or what is it binding to in a signaling role? So this is kind of a major line of work that we're just now, over the past 6 months, starting, is now that we've had the success discovering these molecules, can we more rapidly characterize their function, and can we do that by predicting protein-metabolite interactions? But it's still challenging.
Thank you for a very nice talk, even for somebody with very little knowledge of biology. This was very interesting. I was in particular intrigued by the fact that what you showed in the very beginning, right, that we know only a very small fraction of these metabolites, right? And so one thing that we always talk about in machine learning is generalization, right? So I was wondering, do you think that these few metabolites that we know, are they some kind of good representation of this whole space of metabolites? Do you think there's some kind of blind spots that we have either through the methods that we use to to discover the structure or to even find them? Do you think that these machine learning tools can help us maybe dig into those too? Yeah, your thoughts on this would be appreciated.
It's a great question. I think there's a spectrum of novelty among the unknown metabolites. Some unknown metabolites are very simple, transformations of known metabolites in a way that's obvious. Once you see it, you think, of course, that has to happen. And then others that we've had some success at discovering have been totally unexpected. Chemistry that is not known to occur in mammals at all and required, it certainly wasn't the top-ranked prediction. We had to kind of go down the list and make a few structures before we landed on the correct one. So I think that the models that we have do struggle to generalize a bit, because you can easily imagine that there are other metabolites that we haven't been able to discover simply because they're so exotic we don't even have a foothold on them. I mean, this is a fundamental problem with kind of machine learning in general. So one thing that we're doing now that I think is going to be useful is just expanding the definition of what we're trying to generalize from. So these models that I showed are all, in one way or another, trained on mammalian metabolites. That basically means human and mouse. Plants are known to do a lot of really crazy chemistry, so are bacteria. And so one thing we're working on is scaling this model to all of terrestrial metabolism. With scale comes the ability to use more interesting architectures also. And so we're seeing we haven't actually tested that model in the wet lab yet, but we're seeing some pretty exciting results with that. But I think that might be a way to sort of better generalize to unexpected metabolic chemistry that occurs in mammals and is known to occur somewhere else in the kingdom of life, just not necessarily in humans and mice.
Hey, any other questions? Here.
Yeah, thanks for the nice talk. This may be related to what you were saying right now. So you gave an impressive amount of evidence that the model is somehow, through statistical learning, extracting important features of the chemistry of metabolites. Do you believe that this information can be retrieved from the model? So these models are interpretable in some sense? So you mentioned the functional groups that were recurring at the end.
Right.
Do you believe that you can list, let's say, these important features?
Yeah, I mean, so to be perfectly honest, this isn't something we have worked on, but it's something that other people are starting to work on. Obviously, there's been a lot of interest in interpretability of protein language models, and there's been kind of a nice suite of tools either developed or applied in that setting, and people are starting to use the same kind of interpretability tools to look at chemical language models. I'm aware of like a couple preprints that came out fairly recently that basically suggest that the models learn actually functional groups in a specific order. Like if you look layer by layer, there are neurons activated by specific functional groups. I can't, claim to be an expert on that. It's not our work, but I think it's an interesting line of work for sure. And presumably those interpretations are going to be a little brittle in the sense that models trained on different corpora of structures probably learn.
Do I need to do something in here?
No, wait.
Okay. Thank you very much.
Oh, you should talk with the mic, or
Can I use that?
prefer? Yes.
Because I like to move around, so it's on already.
No.
Let's put it on.
Okay.
Is there
We have to connect to your computer.
Okay, thank you very much for the invitation and for inviting me to be part of the evaluation jury. And thank you very much for ICTP for all the fantastic work that you do for the scientific community, and also for IBM for supporting the next generation of scientists. Before I start with my talk, I want to say, what if we move towards a world where the models that Michael talked about are much smaller, more specialist? What if we have one or two large models or smaller models, open weight models, and we don't need to retrain every time? I was going to ask Michael about the training cost for your models and architecture for these models when you develop these systems. So what if we move to a world where these models are much smaller and the system behind it is very smaller? But before I talk about my talk, I would like you to spend 30 seconds, close your eyes, and tell me a little bit about the future with the best version of AI. If you want a world without AI, that's fine. You could imagine a forest or something without IBM, that's fine. But what is your best version of AI? So close your eyes for 30 seconds, or less, maybe 20 seconds. And imagine a world with AI, not just on your desktop, but everywhere. What would that be? And I'll move around here to see. Okay, so anyone will give me some ideas to start with? Or are you still imagining? Come on, any crazy ideas? What's your favorite world with AI?
I would love to have a sort of private intellectual partner, say, in my smartphone or something like that. Something I can continuously dialogue, have discussions.
And that's as far as by phone. How about you? How about your shopping? When you go and do some shopping or when you eat your meal, what's your best version? How about you, Alessandro? What would be the best world with AI that is safe, let's say, and private and everything? Let's just assume privacy is there.
Yeah, I do think that the most important a world where AI is transparent, open, and reliable.
Democratize, maybe? How about Michael? Away from your work with the chemical world, for your personal life, where would you use AI?
More modest vision of an AI coding assistant that doesn't constantly make mistakes.
So coding, still think about work, which is good. Right, so my version of the AI, and by the way, when we talk about language models, when we talk about ChatGPT, when we talk about, Claude, let's say, or other types of language models, in my opinion, this is the technology of yesterday. It's already done, right? What I asked you about just now, it's about today. It's not even the future. Even quantum and the brain-inspired or all these technologies, in my opinion, these are technologies of today because they are happening. And there are technologies about to emerge. But my version of AI is physical. It's everywhere. You move, let me show you what, I have a missing slide. So my version of AI is physical. So you leave your desktop, you go to the outside world, you go do shopping, you stuck in a hopefully not, in a disaster, and all these devices come to the rescue. Whether it's a small language model, whether it's a collaborative system, whether it's a small model, whether it's a specialist model or adapter, they work together to help you to do what you need to do, still hidden in the background, still private, still safe, still available for everybody with very, very low cost and also low energy requirement. And that's what we've been trying to do in my lab for many years, until the craze about physical AI. I don't know how many people heard about physical AI. How many people heard about physical AI in particular? So, you know, physical AI is the trend at the moment. I mean, besides many, many other things. If you talk to NVIDIA, if you talk to people from you know, other organizations, you'll find, like, in our world, we talk about physical AI because we want to move beyond the text as input towards sensor data, for example, towards contextual data. What if we, for example, support the work that you do in terms of the chemical work that you mentioned with environmental information, about how people live their lives, and then blend this with the biological data and so on. What if we blend that world of the edge AI or the end system, the wearable, with the data center, with the data center in space and so on, and we have continuum of AI going around all of these mediums. So we've been doing this for many years in terms of embedding AI in green spaces, in working with the police, creating platforms, and so on. So what I want to talk to you today is about two main things, or multiple things actually. One is the tiny ML. It's the world moving towards small and specialist models, open weight models, and I call them also collaborative models. The other thing is that it's not enough to talk about the models. It's very important to think in terms of the system itself. You could have the best model in the world, but it consumes too much energy, right? It might not have the right hardware, or it might not have a shared memory. It might make a lot of errors at some points. So the whole system approach is very important, and that's something we try to drive in terms of, like, software, hardware, optimization. I think I have the wrong presentation here. Sorry, I need to move to the. This is my other presentation. Sorry, that's why I don't have the specialist. Can you see specialist? Sorry, this is the other. This is the one. Oh, yeah, that one.
The computer disconnected.
Oh, sorry. Yeah, that's the right.
When you exit the presentation.
Because I have 3 presentations open, and that's why I'm moving between.
Let's see.
Yeah.
Wait.
Exactly. So that's good. Thank you. Yeah.
No, but
No.
use the connection. Okay.
So the famous AI scientist, Jan LeCun, has been talking for some time, and he even established his business, or new business, around the ideas that large language models based on text have reached their limit. And now is the time to move to the physical world and start to understand context. But because I'm not Jan LeCun, we've been working on this for many, many years, but nobody listened to us until, you know, top scientists started to talk about it. In my lab, we have been talking about this for many, many years. Like, what if we move intelligence to the physical world? We created tangible interfaces that can, you know, think and compute. As I said, like smart spaces, pervasive computing, where you have smart edge devices, for example, where we have AI operating in the background, empowering people to do various tasks and learning about, for example, ecology or connecting with each others. And all of that enabled not just with AI models, but also understanding hardware. It's very important to work on the AI side of things and the models, but also understand how can we accelerate AI in a way that is efficient, sustainable, private, and at the same time can fit on the small devices, whether it's for the developing countries or for critical applications like disaster management. So like what Sandro mentioned, I lead the Green Spaces or Green Plus network in the UK. I'm based in Nottingham. where we try to work with green spaces, such as parks and recently organizations that look after amphibians, for example, forests, adventure parks, and so on, where we bring engineers, scientists, AI experts at the high, low level, not necessarily just apps or, devices, but also different way of thinking. Like, for example, green connectivity. How can trees talk to each other and connect them together in order to create a sustainable transport system, for example? And also, I lead the TinyML UK network, which is about to start. And the whole idea here is really to drive the notion of decentralized AI, democratized AI, and again, bringing different communities together. We feel like the community at the moment is very fragmented. You come to one event or activity and people talk about AI, optimization, math, machine learning, deep learning, which is great. But then I go to another world where physicists, for example, talk about neuromorphic, talk about photonics, talk about analog systems or memristors and so on. And then there is the other world of tooling, verifications, security, and in-memory compute, and so on. So how can we bring all of these communities together to come up with efficient, sustainable systems, hopefully small enough? And I work also with the organization called Edge AI Foundation, based in California, where we try to promote the concept of Edge AI. And we had our first Edge AI London event after many years around Europe recently. So the move for AI is really towards contextual, physical, and heterogeneous systems. That doesn't mean we're forgetting about traditional work, biology and other things, but we are enabling new types of collaborations, if you like. And that's why, for this particular talk, normally I talk about TinyML and so on, I started to talk about small, specialist, collaborative, open-weight models. Small, because we can't always think in terms of large models, billions of parameters. Sometimes we don't need the knowledge of the world to do shopping, right? But we might be able to use AI to help us select some items that suit our requirements. But again, for that, we need only smaller number of parameters. And here I'm talking about millions of parameters as opposed to billions or trillions of parameters, if that makes sense. So that's why we need specialists. But not just a specialist, but also adaptive models that can adapt to our needs, personalized, and collaborative in a way that we can bring these devices together that can utilize their resources. Like, for example, I might have a wearable. How many people have wearables here? No wearables in this place? Everybody's worried about privacy? So I will assume there are some wearables somewhere here. One device with 1 megabyte, 2 devices with 2 megabytes, 3 devices, 3 megabytes. If you have 10 devices in this place, you have 10 megabytes of memory, right? And then that's a bigger memory than one individual device. What if you bring that computational power together to enable some type of application or grow the model a little bit to do that? So that's what we call resource sharing. So as I said, we move toward contextual AI, like on-body AI or in-space AI. So when I say TinyML or Edge AI, you need 3 types of components to enable that type of system. You need a computational device, a microprocessor, or processor, let's call it. You need some kind of source of data, sensors, and you need machine learning or some sort of model that can sit on that device, which is not too different from how we do AI in general, but instead of having large data center or a server, we're talking about smaller device. And in terms of the architecture of this TinyML system, you could see there are many variations, whether it's the compression of the model to make it smaller so it can fit on the device, or the deployment of the model, whether it's the inference of the model or retraining of the model, and a collaboration of the model with other things, or finding the optimal model for that type of device. And I mentioned earlier microcontrollers, and again, I don't know how many people are familiar with microcontrollers, but it happens that Microcontrollers actually are small computational devices. You find them sometimes on smart watches. Not all smart watches have microcontrollers that are capable of processing, but we're heading toward that direction. Mobile phones have very powerful microcontrollers in closed systems, right? So microcontrollers enable us to process, to deploy AI models. And in order to do that, we need an instruction set architecture. Like, for example, normally provided by ARM, by x86 Intel, or even open source ones like RISC. And beside the instruction set architectures, which are more like instructions for the processor to execute, there are also other means to do computing within these microcontrollers. So for example, you might have extra accelerators, what we call like neural processing units, or you might have GPU, like a tensor unit, or the latest ones, like the neuromorphic ones that are powered by analog, for example. So there are many variations of microcontrollers, and they are much smaller. They have much limited memory comparing to a PC or workstation, for example. And their energy source is very limited. So these are some examples of some of my favorite microcontrollers. I mean, to be honest, this slide needs to be updated every few weeks, because every week or two there's a new microcontroller, a new type of technology, new processor that we can't keep up with. But the idea behind some of these microcontrollers is that they really advance very quickly, and their capabilities of fitting many transistors, for example, is changing. I mean, I've been told that now we reached to the point where the size of transistor is the size of atom. So we can't go any further beyond that. Some of these microcontrollers have multiple processors, dual processors. The latest Qualcomm one has 4. So you could imagine in such a small device, you really start to be able to do some considerable computation. You can't even run small language models. Some of them are really low cost, like we're talking about a few dollars. Some of the ESP microcontrollers are a few dollars. When COVID hit, we bought about thousands of these microcontrollers, each for like $1, $2, and we created a network of devices running very small models for the contact tracing. So you can't really create a whole lab from these small devices. You could even make them fly. Put some wings on these microcontrollers and they fly and create some kind of drones. And they will have their own AI models, right? And then these drones can talk to each other. So you have a network of drones or collaborative drones, some of the areas that I'm also working on extensively. But if we go back to the concept of making the model small, because I said like we want not just to think about the hardware, but also the model needs to be small to be able to fit in that device. So we can't just take a whole language model and try to fit it in the device. So we need to do lots of squeezing. My PhD students spend a lot of their time on that part, squeezing and squeezing and squeezing, removing this neuron, removing this weight, removing this layer, trying to get the most important parts of the model to stay and remove the others in order to make the model smaller. So there are many, many techniques to compress models, and I will not go in great details, but I can give you very, very quick examples of how we increase the sparsity of the model. Some of it already traditional, like regularization and thresholding and so on that we use anyway. But there are techniques such as pruning, where what I mentioned before, you could really have some kind of algorithm that allows you to remove the least important neurons or layers or part of the model itself. Or quantization, where you reduce the precision of the model. So instead of having 64 or 32 precision, you could have like 8-bit or 4 bits or mixed precision or even 2 bits. So you could have a binary network for some cases. Of course, here you will sacrifice a lot of accuracy, but it depends on the application itself. You could do knowledge distillation. I don't know how many of you have heard of knowledge distillation, where you have a teacher model and a smaller model. One model will teach the other one in a way that the smaller model will learn from the larger model on how to improve its performance. And then there's the idea of AI creating an optimal AI model. So you have the neural architecture search. So you throw some layers, some weights, some architectures into that particular AI model and ask it to give you the optimal AI model for your own task. In fact, people use these days ChatGPT to create their own model. They do this prompt search. There's a systematic way where you could create your own AI model using current language models. But then the story doesn't end there. Let's say you have created this model and you made it smaller, right? How about the sensing part of the system? In our case, we have sensors. I mentioned that tiny ML system should have a sensor, right? So it's not enough to say, okay, I have sensors. I have 3, 4 sensors, and then let them feed their data. I'll deal with it with this small model, and that's it. Sensors can consume all the power if we leave them on. So we need to find a way where we could control it. And we could do, like one of my PhD students was working on what we call layer sharing or parameter sharing across layers, for example. And I'll explain briefly later. But one of the most important things when we think in terms of the whole system approach is that it's not enough to think of sensor feed and the model itself that we have fit, but we need to make that system work for that particular person, right? So for example, if I have a medical device to manage diabetes, if I have a diabetes, I need that to work for me as a person. When you fit a model that is trained on the whole population or large number of populations, that works for that general population. But how about personalizing that model to work for that particular person? And then personalization comes enhanced because it becomes very important, especially for healthcare applications. We could talk here about like to share or not to share, because you could create a generalized model or personalized model. If you create a personalized model, you might not want to share the user's data for, like, let's say, public health research and so on. So it needs to be balanced. But you will find this very often. Someone talked about language models sometimes forget or hallucinate every now and then. And one of the reasons for that is that when we talk to language models, I don't know if you experience this, you ask a question and it tries to guess, like, why can't I sleep early? And it tells you, have you had a meal before? Have you exercised? Have you been tired? So it tries to guess. It gives you very, very generic answer. And then when you ask another question, it will say, no, yesterday I slept at this time. And then I tried to do this at this time. It starts to say, oh, okay, you sleep early, you ate this, but have you been stressed, for example? Again, it tries to guess. So every time it tries to ask you a question to be specialist, so you realize this language model is not specialist at all. It doesn't know much about me, right? It has some kind of knowledge and it tries to guess what do I need. But what if this model knows about you and responds to your need? And that's what we mean by personalized model. So you start with a global model and you make it specialist. You make it more personalized. But one of the issues when you start to make that model specialist, you deal with the catastrophic forgetting issue. Is that you start asking your language model some questions, and by the time you asked many questions, it forgot the initial question. And now it started to talk about your interest in football or the World Cup or something else, rather than your initial question about your sleep pattern, right? And that happens because there are many parameters at play in that model. And it starts to get confused, or it forgets about the initial discussion. The new parameters started to overpower the previous one. So the way to deal with that is that we try to retrain part of the model, what we call continual learning, to make that model respond to your need at this particular time or for this particular task. So continual learning is really one of the very important and emerging research, let's say, where we try to solve this problem of catastrophic forgetting, but also updating the model, making the model adaptive. So you get like general model, you try to update part of it to make it specialist. Maybe you try to train part of the parameters, and then it helps in terms of local adaptation. So for example, you might have a specialist model for non-destructive testing. We do that with some of the, like for example, Airbus and other aircraft organizations, where we try to understand the behavior of machine without opening that machine and so on, or vision problems and so on. Skip this one. And then you might have multiple models at play. You might have audio model, you might have motion model, you might have vision model, you might have chemical model, or other sort of models that are trying to work on that particular task. And then you start to ask yourself, do I need to run this full model with the whole parameters, or are there some ways to share some of these parameters for the common tasks between these parameters? So these type of questions are very important, rather than assuming that I will run everything to the full. And the reason for asking these questions, because I have a very limited budget on that device, I have a very small memory. I can't afford to have any neuron that doesn't contribute to the learning. I can't afford to have overlapping parameters that are in that device. I'll skip this as well. And then when it comes to language models and moving to the physical world, so we have language models and you have the physical world. How do we make this move? I don't know if you have heard of, I'll jump to the open models, open weight models. Did anyone hear about open weight models? Yeah? Do you know roughly what they are? What's the difference between open weight model and ChatGPT, for example? No? I can't tell you, it's okay. So, ChatGPT or Claude, for example, right? They don't give you access to their parameters. They don't give you access to their model, but they give you access to API, so to build your application on top of their service, right? But you can't access their own weights. You can't just take their model and put it on your device. You have to connect to it. You have to connect to that large model. And the reason for that is that that's their own business model. So they are based on language models. If they give to you their model, their business really is in trouble. But if you ask Meta, for example, their Llama models, they share the weights. They have open weight models. They share the weights in a way that you can integrate it into your own model, right? And even take part of that weight and utilize it. Because again, language models are not the core business of Meta, same as other organizations like Google. They have some kind of open weight models. Open weight models are very, very important. If you want, like, for example, to create your own models based on foundational model in the future, you might borrow some open weight models rather than retraining the whole thing from scratch, or borrowing part of that open weight model and use it in the system rather than retraining. So it helps in terms of saving power rather than retraining every time. So yeah, open weight models turn the model into a starting point. So I'll move towards more or different topic that is complementing to the TinyML pipeline. So I mentioned that the software, I mentioned the AI, I mentioned the optimization and so on. What about the hardware, the computation? How can we create an architecture that keeps up with the innovation in AI, that is energy efficient and small, let's say? Again, I'm assuming some of you have heard of neuromorphic, brain-inspired computing. Yeah? Again, anyone heard of, don't worry, I'm not going to ask you directly, but put your hand up if you heard of brain-inspired. We have too many shy people here in the room. Right? Brain-inspired. So neuromorphic is the concept of why don't we come up with new computing architecture that mimics how the brain works, in a way to make it energy efficient, in a way to make it faster, right? And can mimic the way that the brain works. And now, especially in the UK, we have many research attempts at coming up with new architectures, like, for example, analog computing, which might be based on the new materials, which might be based on photonics, for example, to come up with these architectures, sometimes moving away from the traditional silicon-based chips, for example. So neuromorphic computing, I mean, I'm not going to go through all of this, but it's really an emerging field that is trying to innovate in terms of how we compute. And I think it matters to all of us. All of you who are using AI need to know about neuromorphic, need to know about these emerging architectures, because it's not going to stay the same for a long time. Even we hear about a new type of analog computing that is going to change the way we do compute. It might even replace the digital world, which I don't agree on 100%, but it might in the future. And then we need a different way of doing software, a different way of doing AI, different way of interacting with these systems. So it's really of our interest, whether you are a computer scientist or someone who was developing AI models or utilizing AI models. And I have to mention here, IBM has been a pioneer in neuromorphic. I mean, currently there are multiple IBM systems like True North and North Pole, but personally in my lab, we are using some of the current simulators, which enables us to understand the analog world, because sometimes it's very hard to have actual neuromorphic or analog device, because it's still developing. So we use simulators to access that. Just to give you an example of a project that we are working on at the moment, which is funded by our engineering council in collaboration with other disciplines, as I said, in material and physics and photonics, where we try to utilize analog computing alongside TinyML. So our approach at the moment is to see if we can move toward analog computing in the near future. But the issue is that, as I said, analog computing is still progressing, still in its infancy. Like, for example, you could see here on the right side some of the in-memory compute or a new type of memories called register memories, which are analog. These are not very easy to create. So what we proposing, or what we propose in one of our papers, is that how can we use analog computing to accelerate the current digital compute? So until analog computing is mature enough, we enable it. We create this TinyML that hands the features, for example, from the model to in-memory compute, and see if we can save some time and reduce latency. And the whole focus here is on energy efficiency. So again, I will not go in details. Another thing to mention, that we've done the initial paper, which is the hybrid digital analog, and at the moment we're trying to make it adaptive in a way that it can update. I know I'm running out of time, so I'll try to go very quickly to say neuromorphic that I mentioned, brain-inspired is one type of architecture that goes to the device itself, but there are other emerging architectures as well that we are utilizing. One particular system or architecture that we are building in my lab in collaboration with other researchers, we call it node learning. A system that doesn't require server. It doesn't rely on any central point. All the nodes work together. It can be one, it can be two, it can be three, it can be millions that are interacting together. And if you remember my initial image on disaster management, it's all to enable these devices to work collaboratively to achieve a task. Imagine, for example, you have a cluster or a processor. You cut it into 4 pieces, okay? One computational device. It has an AI model. You take these parts in different places, and the model still works the same way. Still operate on these different devices. So here we're not talking about traditional distributed system. I will talk about this tomorrow in my lecture in the morning, but we're talking about AI sitting between these devices and being updated continually. And that model is floating between each others. And you could share data. I mean, data is not a good idea because of privacy issues. You could share gradients, and that's another issue. But you could share parameters, features, layers, prediction, uncertainty, but also you could share layers, and as I said, and models. And that will enable the move towards heterogeneous, physical, and contextual AI. One thing to mention, like for example, why I keep talking about the idea of heterogeneous. Why do we need heterogeneous systems, even within one device? Imagine, like for example, you have this drone, and it has the processor. So there's more to drone computation or movement than just the computational part. There's also the mechanical movement. And then you could have another device there. And you might have another device to do some actions, which means you have different systems at play here, talking to each other. What if we think about that architecture and create a new chip that is heterogeneous, able to work with all of these devices? And then, I mean, there are many, many things at play when it comes to heterogeneous physical systems. So I will not go very long on these. So I just want to deliver this message, is that there's a lot of discussion about large language models. Again, technology of yesterday. I'm talking here about technology of today, small, specialist, collaborative, open weight model. Maybe technology of tomorrow will be about energy harvesting devices that can have their own energy connected to AI devices, maybe things I have never thought of. So we are at the stage where we have these type of models that are operating. We just need to take it to the next level. And we need to ask questions about, if we can make it a specialist that doesn't forget or forget a little bit, unlearn, distribute, and use new type of substrate to enable analog computing. That's it. Thank you. You could use that.
Oh, we could use this as another one. We have another one. Yes. Thank you very much, Ayman, for this beautiful talk. Questions or comments?
I have a curiosity. So in the node learning approach that you illustrated, so how do you treat single node failures? Because you don't have anymore a controller node that regulate the flow of information.
Very good question. The system has to be very robust and elastic. In a way, it doesn't rely on one node or two nodes, so it can work by itself. So that's why, I mean, in one of the images or illustrations, I showed this. The idea is that you still have edge AI, you still have the device. That device is aware of other potential connectivity. If there's no other connectivity, it acts. There's another device, it acts and works with them. Another device disappears, it goes back to its initial state and work again. So there's assumption that any device might disappear at any time.
But so with respect to the standard federated learning approach, in this case, are we duplicating more model information? Because I see that you put there that we have a local model and a shared model on each node device.
The idea is that they complement each other. So each of them is trying to do this particular task. Like, for example, the scenario, and I'll talk about this tomorrow, like missing person search and rescue. So one person or one node will search for that missing person. Two will also search, but they will do it faster. You have three, they will do it even faster, right? So if one of them disappeared, you go back to the two persons kind of scenario. So the system has to be robust and elastic enough in a way that it doesn't rely on particular node. It's the system that should carry the burden rather than one particular one. And because also we have an issue with the network. The network might disconnect at any point.
More questions? Anything from the students, from the participants?
Thank you for this very nice and broad talk, the vision. So I was wondering, it's a field that I don't know much. What are the specific constraints when you have a highly distributed network of agents like this on security. So already when we're talking about a lot on data safety, privacy, when we're discussing these located large language models, what are the differences in terms of privacy and security when you distribute the data like this in very large networks of agents?
We're not distributing data. Remember, data, the current way is to distribute weights and maybe even closed models. So we try to avoid exchanging data or even gradients, and I mentioned that earlier. But even with that, there are possibilities to, reveal some private information. You want the short answer? We don't know. Because the reason for that is that it's an emerging field. We're just trying to see if we can make it work as a starting point. And of course, we have to be concerned about like not just the security, but what if we have unknown node joining the system? How do we verify, and how do we know that we only trusted nodes that need to be involved? There are so many questions when it comes to this, which I'm hoping that once we start working on this at scale, that this will be one of the mainstreams to look at security, safety, and also, I talk a lot about cyber and physical security. You'll find many organizations think about their cyber security kind of systems, but then I tell them, how about physical security? Because these devices can squeeze and go inside the building without you noticing and do things. So we need always to think in terms of cyber and physical security.
Thank you very much.
Thank you so much. I have a question. It's more maybe a curiosity driven by my own research. But given that we are speaking about the collective behavior of these multi-agent systems somehow, have you ever wondered which is the minimal model size before the model are aware of being part of a collectivity? In other words, how big the model should be to realize to be part of a multi-agent system?
So, when I talk to my students, when they design the system, we always ask for the smallest models possible. But our limitation is really the memory. It depends on how much memory you have. If you have 1 megabyte, your limit is 1 megabyte. In fact, it's less because you have other overheads and other things to consider. So as small as possible, but you look at the memory, RAM and flash, and see what are your limitations, and your model will be within this constraint. You can't fit a bigger model than the memory you have.
Okay, because I was afraid of the fact that if the model is too small, it's not able to realize that it's part of something bigger and acts as a single model.
Are you attending the lecture tomorrow?
I will, yeah, I will be there.
Because we'll talk about the global model and
great.
Yeah, the local model, and how we try to make them work.
Actually, a question from online, if I can read it to you. This is a question from Mohammed Asib. The question is about Apple tags. Can we say that Apple tags are an example of open weight models or not node learning? If not, can we have a real-world example where they are implemented and working in the present?
So the question is whether Apple has an open weight model?
Yes.
I'm not too sure.
No, Apple tags. The Apple tags.
What is that? Apple tags?
Yes.
On the models? To be honest, I don't use Apple's models. Yeah. We could ask ChatGPT.
Of course. Okay. I guess if there are no other questions, let's all thank Ayman again for the beautiful talk. And you're all invited, I guess, on the terrace, right, Adriana? We're on the terrace, right? It's on the terrace for refreshments, so we're all invited to join us.